Okay, guys, it's time to start rolling. So I'm going to get after it. Chapter 9 in your textbook is about muscles and muscle tissue. So I'm going to throw this disclaimer into this particular video. Um, it's always a good thing to watch these lectures or attend these lectures, watch the recordings, but it's not really an excuse to not read your book. So please don't fall into that trap. Students who do both, who read their textbook and either attend these Zoom lectures or watch the recordings, always do better than students, obviously, who don't do either, but even students who do one or the other. So I'm going to do an introduction about skeletal muscles in relationship to other types of muscle tissue, and then talk about the bulk of this chapter is just about skeletal muscle tissue. But I'm going to call it my first whiteboard. Is so I'm just going to write muscle and muscle tissue. And there are three types of muscle tissue. First one is skeletal muscle. That's what you and I think of when we hear the word muscle. Skeletal muscle is the stuff that's attached to our bones and enables us to move our bodies, move our appendages. There's also, let's do some general characteristics about skeletal muscle. First, physically, these have really long fibers. So they're really long cylindrical cells within the muscle tissue itself. These have striations. Just think of stripes or banding patterns. When we look at these cells in this tissue under a microscope, there are visible stripes or banding patterns. This muscle tissue is voluntary. That means we have conscious control over the contractions. If you want to raise your arm or flex your elbow, you can do that. So it's voluntary or consciously controlled. The cells, the individual muscle cells, are multinucleate. That means there's more than one nucleus per cell. And I'll explain later in the chapter how that occurs, how that comes about. So as I said before, the location of this stuff is all over the body, and this is what we find attached to our bones. And finally, each cell, each individual cell must be neurally stimulated in order to contract. So each cell has, correct my spelling there, each cell has its own nerve ending. Without direct neural stimulation to each cell, that cell is unable to contract. Second type of muscle tissue is cardiac muscle. Say a few things about that. I'll try to be consistent. Cardiac muscle also is striated, has those banding patterns or stripes, but it is involuntarily controlled. We do not consciously control cardiac muscle contractions. The cells can have 1 to 2 nuclei per cell. So it's possible for these cells to have more than one nucleus, but it's rarely, if ever, more than 2. So it's just 1 to 2, whereas skeletal muscles can have many nuclei per cell. Finally, the location. We only find cardiac muscle in the heart. And every single cell does not need its own neural stimulation in order to contract. Cardiac muscle has the ability to transfer electrical stimulation from cell to cell. Once one cell is stimulated, there is an ability for that electrical impulse to be transmitted from cell to cell. So I'm just going to write every cell doesn't need neural stimulation. And finally, the third type of muscle in the human body is smooth muscle. Smooth muscle gets its name because it is not striated. It does not have a banding pattern or stripes when we look at it under a microscope. It's also involuntary. We do not have conscious control over the contractions, which ultimately means it's regulated by the autonomic nervous system. One nucleus per cell. So it is not multinucleate ever. The locations that we find smooth muscle are in the walls of organs. Think the digestive system, the GI tract, and the respiratory system, in the walls of organs and vessels, in our blood vessel walls as well. And this tissue, every cell also does not need neural stimulation. So I'll just write that quickly. It also has the ability to transfer an electrical impulse from cell to cell. Now, from this moment forward, pretty much the rest of the chapter until the very, very end, the chapter says something about smooth muscle. 95% of chapter 9 in this textbook is about skeletal muscle. The reason is because you will learn more as a student about cardiac muscle when you study the cardiovascular system. And you'll learn more things about smooth muscle when you study the organ systems that contain smooth muscle. But there is a few pages of smooth muscle information at the end of the chapter. So this one is largely what we're going to talk about from this point forward, and a little bit smooth muscle at the end. Before we jump head first into what a skeletal muscle structure is like and all the components, we do need to talk about general characteristics of muscle tissue. General characteristics of muscle tissue. And I'm just going to list these terms and speak about each one individually briefly. The first one is excitability. So muscle tissue exhibits it's trying to write ability, not excitatory. Excitability. That means that muscle tissue and muscle cells have the ability to receive and respond to an electrical stimulus. They're excitable electrically, neurally. The next general characteristic is contractility. We take some of these for granted. We just know they exist. But what contractility means is that the cells, once stimulated, muscle cells have the ability to shorten or contract. And what we're going to see is that ultimately gives the ability to do some sort of work. So the cells can shorten when stimulated. That's what contractility means. Extensibility. That means they have the ability to stretch. This tissue in these cells can be stretched. And finally, what's related to that extensibility is elasticity. Get my eraser out here again. Elasticity. That means they have the ability to recoil or snap back to their original shape or length after they've been stretched. So the three E's and the one C are general characteristics of muscle tissue. and general functions of muscles in general are muscle tissue produces movement in the body. That's its main function, but there are others as well. Especially skeletal muscles, help maintain posture. So we get movement, we get the maintenance of posture, stabilizing joints. As we learned in the joints chapter of the book, what fortifies or strengthens a joint more than any other factor is the muscle tone around that joint. Muscle tissue and muscles can also generate heat, or produce heat. When they contract, heat is produced, can be used to regulate body temperature to some degree. Protection. I'm just going to write protect organs. So your thoracic cavity is protected by your ribs, your rib cage, your heart, and your lungs. But your abdominal cavity is largely unprotected by any bone protection, and your abdominal muscles all the way around, laterally, anterior and posterior, are protected largely by sheets of muscle. And finally, muscle tissue and muscle cells have the ability to store nutrients. ATP is an energy source that can be stored a little bit in muscle cells, as well as glycogen. Most, particularly skeletal muscle cells, store glycogen, which is just a stored form of glucose. Okay, now we're going to take off. And from this point forward, like I said on our first board, every time I say the word muscle, from now on, I'm going to be talking about skeletal muscle tissue or skeletal muscles, okay? Those are the things that are attached to our bones, and we have voluntary control over them. So I'm just going to write a huge header here, skeletal muscles. First, to remind you, every muscle cell needs to have its own nerve ending or neural stimulation in order to contract. So a bit of language here. An individual skeletal muscle cell we also refer to as a fiber. The reason is because the cells are really long and cylindrical like this. Because of their shape and their length, if you hear or read fiber, just think that's an individual skeletal muscle cell. So having said that, each fiber or individual cell must have its own nerve ending in order to have a chance to be able to contract. These skeletal muscles also have a very rich blood supply. There's typically one major artery leading to a major muscle, and then that artery branches, usually enters right around the central portion of the muscle, and then branches profusely throughout the muscle to provide a rich blood supply. So they are highly vascularized. They are also highly innervated. So motor nerves supply skeletal muscles with the innervation they need in order to contract. There's also things called glycosomes within each muscle fiber cell. A glycosome is just a small organelle. It's like a granule inside of each cell that contains glycogen stores, which are energy storage. Glycogen, you should have learned what glycogen is back in Bio 1. If you have forgotten, glycogen is just a stored form of glucose. The individual glucose molecules are just linked together to form a molecule called glycogen. And finally, I'm going to write, before I leave this board, I'm going to write the word myoglobin. We find myoglobin is actually a protein that turns red when oxygen is stored inside of it. So this is a pigmented protein that has the ability to bind oxygen. Just going to write binds O2. Like hemoglobin in our blood, myo just means muscle. Myoglobin is a protein-based structure that oxygen binds to, and when it does, it turns red. So myoglobin turns bright red, just like hemoglobin does in our blood, when oxygen is bound to it. Okay, now macroanatomy of a muscle. Macro means we can see this with most of this with our eyes. So muscles themselves, an entire muscle, begins with the structural unit is a fiber. So a muscle fiber, we just said, equals an individual cell. Each fiber, muscle fiber, is wrapped in a thin, delicate connective tissue called endomysium. Endomysium, that's M-Y-S-I-U-M. Endomysium is a thin, delicate connective tissue that wraps around each individual fiber. Now, if we take a bundle of those fibers, a bunch of those wrapped fibers, and we group them together, I'm just going to write a group of fibers then gets wrapped in a slightly thicker, coarser connective tissue called perimysium. Perimysium. Perimysium is a little bit thicker and coarser than endomysium, and it wraps a whole bunch or bundle of individual fibers. So individual fibers are wrapped in endomysium. A group of fibers then gets bundled together and wrapped in perimysium. What we call a bundle of fibers that is wrapped in perimysium, we call that a fascicle. F-A-S-C-I-C-L-E. So a fascicle is just a bundle of muscle fibers wrapped in this connective tissue called perimysium. Then, if we take more than one fascicle, more than one bundle, and wrap them in a really thick, coarse connective tissue, that makes up the entire muscle. So an entire skeletal muscle is really several fascicles wrapped in a very coarse connective tissue called epimysium. Epimysium is the outer covering of the entire muscle. So the macroanatomy, the organization of a skeletal muscle is we have an individual muscle fiber, that's just an individual cell. We wrap that in a light, delicate connective tissue covering called endomysium. Then if we take several of those wrapped individual fibers and we bundle them together and we wrap those in a slightly heavier connective tissue called perimysium. So the green is perimysium. And what we've created is a bundle of muscle fibers called a fascicle. Okay, now if I take several fascicles, I'm going to do this quickly. There's one bundle of muscle fiber cells wrapped in perimysium. Now I'm going to make another one right next to it. There's another muscle bundle fiber wrapped in perimysium. So I've got two fascicles now. I'm going to do a third. Wrap it in perimysium. And if I take all three of those fascicles and bundle them together into one huge structure, and I wrap it in epimysium, what I've done is created an entire muscle, an entire skeletal muscle. So that's the macroanatomy of how a muscle is arranged. It's just long cylindrical fibers wrapped together in a bundle. Then we take several of those bundles, wrap them together, and make up an entire muscle. Briefly, I'm going to talk about the attachments of skeletal muscles to bones. So we know that skeletal muscles are the type of muscle tissue that attach and move our bones. All skeletal muscles have attachments in at least two places. Just think one end of the muscle and the other end. Oftentimes, there's more than two attachments, but they must have at least two attachments. So the end or ends where there is no movement when the muscle contracts. So I'm just going to write end where there's no movement during contraction. During contraction. We call that end the muscle's origin. So the origin is the point of attachment to a bone where, when the muscle contracts, there is no movement. The end or ends where there is movement when the muscle contracts, just right during contraction. We call that end or ends the insertion. So really quickly, classic example. Your biceps brachii muscle, that's in your upper arm, that when you bend your elbow, you make a muscle, you contract that muscle. The points of attachment of that biceps brachii muscle, the origin is up at your shoulder area. The insertion is down in your forearm on the radius and ulna. When that muscle contracts, it's your forearm that moves. So the insertion end of the biceps brachii would be the radius and the ulna. The origin, your shoulder, when you flex your elbow, your shoulder does not move. So the origin end is the scapula up on your shoulder. That's just a classic example. Now let's look at microscopic anatomy of a muscle fiber, an individual muscle cell. And it's going to look an awful lot like the macroanatomy. It's just basically small tubular threads bunched together to make bigger structures, bunched together to make bigger structures. So skeletal muscles arrangements, both microscopically and macroscopically, are basically just long cylinders wrapped together to form bigger bundles, wrapped together to form bigger bundles, yada, yada, yada. Think of like small PVC pipes at Home Depot. When they're sitting on the rack together, there's a bundle of them that can be wrapped together And if we take several of those bundles and wrap them together to make a bigger bundle, that's what a muscle really looks like. In order to effectively examine the microscopic anatomy of a muscle fiber, an individual cell, we need to build a vocabulary library. And this is language that your book uses, and this is language that I'm going to use for the whole rest of this unit, this chapter. So what I'm going to do now is just build a glossary or a dictionary for us. I'm going to write the words out and then tell you what they mean. Their written definitions are in your textbook. The word sarcolemma just means the plasma membrane of an individual muscle fiber. So you know that a muscle fiber is just a cell. So it's just the plasma membrane of the cell. So going back to our first page, the endomysium, that connective tissue, that delicate connective tissue that wraps around an individual muscle fiber, would be on the outside of the sarcolemma. This is the plasma membrane of an individual fiber or cell. Sarcoplasm just refers to the cytoplasm, everything that's inside of the cell. In the cytoplasm of a muscle cell, remember, that's where we find those glycosomes, bunch of, I'm just going to say organelles, fluid, glycosomes that we mentioned on a previous page, the myoglobin, those proteins that bind oxygen and turn red, is also found in the sarcoplasm. This term is probably new to you. Myofibrils. It's important you understand as best as possible what a myofibril is. It's a specialized organelle inside of an individual fiber or cell. It's long and cylindrical, and it contains myofilaments. Okay, I'll define myofilaments in a moment, but they're just long tubes, basically. We call them specialized organelles, but they're long tubes that contain myofilaments. Each myofibril can contain hundreds to thousands of myofilaments. They take up most of the volume, the inside volume of a muscle fiber cell, these myofibrils. So what is a myofilament? These are long, thread-like, contractile proteins. That's supposed to be an I. Long, thread-like, contractile proteins. I'll try to draw a picture after we define some of these microscopic structures. So they're very small, thin meaning small, very thin, rod-like structures inside of the myofibrils. That's what a myofilament is. And there's different myofilaments. There's 3 to be precise. I'll number them. 1, myosin, myosin. We call this the thick filament. Actin, actin, that is known as the thin filament. And the third one, some books refer to it as titin, T-I-T-I-N. That's known as the elastic filament. We're going to mostly talk about numbers 1 and 2 in this chapter. I will mention titin or elastic filaments when it's appropriate to do so. These really these help the muscle recoil to its original structural length. All right, let's continue our glossary. So we've done sarcolemma, sarcoplasm, myofibril, myofilament. Next term is a sarcomere. I'll define this term and refer to it later. This word basically refers to this term, one contractile unit within a myofibril. I'll write this one out. One contractile unit of myofibrils. Sorry, myofilaments. So we have repeating series of myofilaments within a long myofibril. And the word sarcomere refers to just one unit, contractile unit within that structure. So back in our previous page, we talked about the myofilaments, myosin, actin, and titin. What is myosin really? We know that it's the thick filament. Here's what that means. It's basically a protein-based structure that has globular heads on the end of it. So it's a long polypeptide chain. That just means a chain of amino acids that has a globular head and a long chain tail. So I'm going to draw a picture of it. There's a globular head and a long chain tail. So this is how I typically, and most books typically, draw what a myosin filament looks like. So that's just a globular head. And this long chain here is called the tail. It's thinner and longer. Really what this is a chain of amino acids, a polypeptide chain that looks like this. And then the amino acids create this globular structure, protein-like structure at the end. It's just easier to draw it the first way. These heads, they form cross bridges with actin during contraction. That will make more sense when we go over the process of how a muscle contracts. I'm just going to write form cross bridges with actin during the process of contraction. The heads also bind ATP, the molecule adenosine triphosphate that we talked a little bit about at the beginning of this course. I'll mention some other things about it while we are in this chapter that are appropriate for you to understand how a muscle contracts. Next term is actin. Let's just describe what actin really is. We know that we've already identified it as the thin filament. Actin is basically just a long, skinny polypeptide chain. I'm just going to write long, thin polypeptide. Technically, it's considered a protein. Long polypeptide chains become proteins when they take on secondary structure. So, thin filament, long, thin polypeptide chain, and it has or possesses active sites for myosin to bind to it. There's something called an active site on actin that this head of myosin can actually bind. They bind together, stick together. Some more protein words. Tropo. These are just funky words. They're not difficult concepts. Tropomyosin. Believe me, I'll explain these in great detail in pictures after we define what they are in words. Tropomyosin is a polypeptide strand. Think really short somewhat short amino acid strand, just a chain of amino acids, polypeptide strand that blocks active sites on actin. Troponin, TRO, and these sound the same, but they are not. Troponin is just a really short polypeptide structure. It's a 3 polypeptide complex. We don't even call it a chain. That is attached to tropomyosin. And this is the important part, binds calcium ions, Ca++. Finally, I'm running out of space here, but that I already said pretty much what I need to say about tighten the elastic filament. Just it helps to recoil after stretching. So I'm just going to write the word recoil. I know this is boring and it's just defining terms, but we need to understand these terms in order to understand how a muscle contracts. The sarcoplasmic reticulum sounds a lot like endoplasmic reticulum. And structurally, somewhat it is like the endoplasmic reticulum in other cells, but specifically in skeletal muscle cells, the sarcoplasmic reticulum is a set of tubules, tubular structure, like smooth ER, smooth endoplasmic reticulum. Its basic function is it stores calcium ions. So it also wraps around the myofibrils within a muscle cell. Okay. Just what? I have two more terms, and then I'm going to get on to describing how this is all set up and how a muscle contracts. T-tubules are specialized structures. That's just short for transverse tubules. These are elongated tubes, or they begin with invaginations of the sarcoplasm, the plasma membrane of a muscle cell. They're invaginations of the sarcoplasm that run toward inside of the cell. Fiber, same thing. Their function is they conduct electrical impulses to the interior of the cell. I'm going to draw a picture with all of these things on it to help you understand this glossary, these structures that we've just defined. Inside the fiber. And finally, the word triad. Tri means three. So triad just means there's three structures involved. And what a triad is are two terminal cisternae of the sarcoplasmic reticulum plus one T-tubule. Oops. So we have 2 plus 1, 2 terminal cisternae plus 1 T-tubule equals a triad. What is a terminal cisternae? A terminal cisternae is just a bulging section of sarcoplasmic reticulum that has large stores of calcium ions in it. Okay. Now, in order to be able to sort of wrap your head around all these terms, these crazy terms that we've defined, I'm going to draw the inside of a muscle fiber cell and try to relate to you what's going on with these structures. So I'm going to draw one large, I'm going to exaggerate how thick it is so that we can fit all these things in here. So a muscle fiber or an individual cell is long and cylindrical, and it has 2 ends. I'm going to draw some of these arrangements in here for you. First, there's a repeating pattern of myofilaments. If it says this is one fiber, one individual muscle fiber cell, inside of here are little tubes that take up most of the space. Each of these little tubes is a myofibril. A myofibril is a long tubular organelle that contains myofilaments. So contains actin, the thin filament, myosin, the thick filament, and titin, the elastic filament. Now, if I take one of these myofibrils and I blow it up, remember, this is just one cell that we're looking at here. But if I take one of these tubes and I blow it up, so this is a myofibril. What's inside of here are regular arrangements of these myofilaments in a banded pattern, in a very specific pattern. And how most textbooks diagrammatically show this, what I have in red are thick filaments. So these bars are myosin, thick filament, and they have those globular heads at the end. They're clustered in groups, sort of like if you put a bunch of golf clubs into a golf bag and the heads are sticking out and the shafts, the stems are all lined up near each other. That's what myosin looks like inside of a muscle cell. So those are myosin molecules, these things. Actin, I'm going to draw in yellow, is arranged something like this. Between those myosin heads are the thin filaments actin, and they're connected to each other like this at the ends. But I'm going to label this actin. That's the long, thin myofilament. Now, what I've drawn for you basically is one sarcomere. It's one contractile unit, but this keeps repeating over and over in both directions of this myofibril. So I'm going to quickly show you there would be more actin molecules attached here and more, then another set of myosin molecules here. And they're attached by this bar called the M line. It's just a protein structure. And there would be more myosin molecules here. This repeats hundreds to thousands of times throughout one myofibril, thousands to hundreds of thousands of times. And then it just keeps repeating in both directions. We would get more actin, more myosin this way and this way. But what I've drawn for you right here is considered one contractile unit, and it's actually two sets of actin and one large double-sided set of myosin. So I'm just going to label this one sarcomere. And just to remind you what that means, that's one contractile unit. And it's this repeating, overlapping sets, this repeating pattern of myofilaments, actin and myosin, throughout the myofibril that gives muscles their striped appearance like this, those striations. What we call those stripes is really just microscopic, the repeating banding patterns of the overlapping myofilaments. So that's what's going on deep inside of a muscle cell. We've got tons of myofibrils inside of a muscle cell, all of these tubes, and then within each myofibril are these repeating banding patterns, contractile units of myosin and actin. Okay. Now the bulk of understanding, I'm almost finished with today's lecture, but I want to leave you with the beginning of how a muscle cell contracts. And it's called the sliding filament model of contraction. Might not get through the entire story on this lecture. I'm going to cut to the chase and then detail it for you step by step. If I go back to my previous picture, the sliding filament theory says this. I'm just going to draw one myosin molecule and I'm going to simplify it, only put one head on it. And I'm going to draw one actin molecule. And it looks like, actin really looks like a chain of beads or a string of pearls like this. All right, on this, let's label these actin. I'll label this myosin. The goal of muscle contraction, of getting a muscle to contract, is getting this myosin head to reach up, grab, attach to actin, bind to it, and then pull the actin molecule this direction toward its M line, structural M line here. So I'm going all the way up to the top of this picture. The goal is for these myosin heads to reach up. Let me get my black pen back, you'll see it better. Reach up right here, attach to actin, and then the myosin head pulls the actin in this direction. And these myosin heads would reach up, bind to actin, and pull these actin molecules in this direction. They all go toward those vertical M lines, okay? So if we do that several times, here, 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 and we do that thousands of times down the entire length of a muscle fiber and therefore muscle, you can see why a muscle would get shorter. We're shrinking this distance thousands of times all the way down the muscle. So that's really the goal, the end of the story of how a muscle contracts in simplified form. Now I'm going to start with this sliding filament model of contraction. And in order to do that, I'm going to show you how a nerve cell is associated with a muscle cell. Everything in red is an individual muscle fiber. So this is an individual muscle cell called a muscle fiber. What's in black up here is the end of a neuron. It's called the terminal end or the axon end. We haven't studied the nervous system yet, so I don't expect you to understand exactly how the nerve cell works. But we do need to learn how, we call this the neuromuscular junction, because it's just the point at which the nerve cell, the neuron, which the nerve cell, there's no physical contact there. So when I say connection, you can see that there's a space there, but this is the neuron or nerve cell that's going to excite this muscle cell. So the neuromuscular junction is just the area of where nerve cell meets muscle cell, even though there's no physical touching or connection. So this neuron, when it fires an electrical impulse, that electrical impulse travels down the axon, which is this stem, and it gets to the end of the axon, which we call the axon terminal. Okay, so an electrical impulse of the nerve cell travels down the axon terminal. When it reaches the end, this terminus end, pores on the membrane open up. So I'm just going to draw holes here. And calcium ions that are out here in the extracellular fluid influx into the axon terminal. So we have an electrical impulse, calcium influx into the cell. Inside of here, this nerve cell, are these bubbles that contain a chemical called neurotransmitter. It's stored in these vesicles, these bubbles. So neurotransmitter, this chemical that's stored in a vesicle, when calcium binds to this stored vesicle, it tells it to migrate to the edge and release its chemical, its neurotransmitter, into this space. That space there is called the synaptic cleft. The reason it's called a that's a T. The reason it's called a synaptic cleft is because we call this entire neuromuscular junction a synapse, S-Y-N. And so we call that space the synaptic cleft. I'll go through this really quickly at the end. Now, on the end of the muscle cell, I'm just going to erase and redraw this, because in this indented area of the muscle cell are little spots. So this whole indented area that I've drawn, I'm reinforcing it in dark red. This depression area is called the motor end plate of the muscle cell. All of this language your book uses in order to explain this process. That's supposed to say motor, M-O-T-O-R. So the motor end plate has receptors on the end of it. The receptors I'm going to highlight in blue. So back to the original part of our story. An electrical impulse travels down the neuron, the axon of the neuron. When it gets to the terminal end, it causes calcium ions out here to flow in. When they bind to these vesicles, these bubbles that store neurotransmitter chemical, the binding sends that vesicle to the edge and it releases its neurotransmitter chemical. Now that neurotransmitter has a fancy name, that chemical. It's called acetylcholine. That's just the specific name of that chemical. So if you hear me say acetylcholine is released, it just means that this chemical, this neurotransmitter, is released into the synaptic cleft. That acetylcholine diffuses across this space and binds to the receptors on the motor end plate. So right here, that acetylcholine, that chemical binds to receptors. When that acetylcholine, that neurotransmitter, binds to receptors on the motor end plate, it causes the muscle cell to generate an electrical impulse along its sarcoplasm, its plasma membrane out here. Okay, that's the first part of this story, and I'll repeat this. I'll start here on my next lecture. Okay, so really quickly, the sliding filament theory model of contraction, we're explaining how a muscle cell, a skeletal muscle cell, contracts. Step one, neural impulse down the axon to the terminal. Calcium flows in, causes this vesicle with acetylcholine in it to migrate to the edge and release its acetylcholine into the synaptic cleft. That acetylcholine binds to receptors on the motor end plates of the muscle cell, and that generates an electrical impulse on the sarcoplasm of the muscle cell on the edge, the outer plasma membrane. I'm going to pick up here next time and then describe or explain what happens when that electrical impulse is generated along the edge of a muscle cell. Okay, so I'm going to cut my recording here. So we were